Can you hear me? ഹലോ. പിന്നെ ഇവിടെ ഷെയർ ചെയ്യണ്ടേ? ഷെയർ ഓപ്ഷൻ ഷെയർ ഓപ്ഷൻ കിട്ടില്ലല്ലോ. മോറിനതാണോ? മോറിനതാണോ? മോറുണ്ട് ഷെയർ. യെസ് യെസ് യെസ്. കണ്ടിന്യൂ. കട്ട് ചെയ്യാം. ഇപ്പൊ എന്നെ ഷെയർ ചെയ്തിട്ടുണ്ട്. ഇതാണോ? അല്ല, ഇപ്പൊ എന്റെ സ്ക്രീൻ ഷെയർ ചെയ്തിട്ടില്ലേ? അല്ല, എന്റെ മ്യൂട്ട് അതല്ലല്ലോ. ഓഡിയോ ഒക്കെ നോക്കി കേൾക്കുന്നില്ലേ? കേൾക്കാമോ? കേൾക്കാമോ? കേൾക്കാമല്ലേ? Good evening, students. I hope my audio is clear, and I hope that you can see my screens too. First of all, let me thank Almighty for this opportunity, in the sense to share the experience and knowledge with you students, and for the well-being. In fact, for forthcoming examination is going to be help you a lot. So, in that way, I thank God. Second, let me appreciate this group, their positivity to make you study in the last moment and trying to go deeper in the subject and make you pass, get a good marks in your examination for their hard work, their enthusiasm, their positive vibes. So I appreciate that. And furthermore, let me congratulate and appreciate the students too for opting this app, this group, for your further studies too. In fact, when you go through a number of experiences you had for the last 1 year, you went through a lot of experiences. You were able to study some topics you were not able to understand. And as I showed the screen, you are not ready to give up. And you are always trying to say it's possible. And if you don't give up, then you know what will happen? You go up. So may the Almighty help you to go up, come out in flying colors in the forthcoming examination. So let me go through the topics one by one. First of all, let me tell you one thing frankly, this is a revision class. So we'll be not going so deep, but we'll try to touch almost all the important points in the topics what I'm going to teach you. And earlier, maybe in last few days, the organizing group was asking you about the feedbacks on which topic has to be taught. And as per that, as per the feedbacks, let me be teaching you on these topics. And first of all, a number of students have asked about oxidative phosphorylations and electron transport chain. So that topic we'll cover first. In that, a small 3-mark question or 5-mark question can be asked, short essay or short answer can be asked. Then, as the students ask, I'll be teaching you about the beta oxidation and then about HDL metabolism. Today, we'll be trying to cover these topics in the stipulated time. So let me start with, I think the screen is not moving. Yeah. There are some technical issues. Hope you're able to see these ones. So today I'll be discussing about, as per the feedback, I'll be teaching you on oxidative phosphorylation and substrate level phosphorylation with high energy compounds, electron transport chain, chemiosmotic theory, uncouplers. These all comes from the group called as biological oxidation. And then about beta oxidation, and then about HDL metabolism. These topic I have been taken since this important topic. At the same time, a number of students have requested to have a revision class on these topics. Okay. First of all, let me go to the topic of oxidative phosphorylation. There comes a word, oxidative phosphorylation and substrate level phosphorylation. First of all, what is oxidation? You all might have studied in your +2 class about that. Just see, my point is always try to understand the basic concepts. Then it will be very easy for you to understand the other things and study well and prepare well and write well. You'll be able to understand and you'll be able to memorize. You'll be able to reproduce if you understand the concept. So I'll always go for the basics first. First of all, what is oxidation? It's removal of electrons or removal of hydrogen, removal and addition of oxygen. These all comes under oxidation. And in living system, any breakdown process is oxidation. So when we say biological oxidation, it's nothing but it's about catabolisms. Okay, breakdown process in the body we always call it as oxidation, and just synthesis process in the body, anabolic process will be reduction. So whenever there's an oxidation process, then the removal of electron, somebody will be there to accept it. So that's getting reduced. So it will be always coupled with reduction. So this is the basic concept. Always you should know what is oxidation means. This I need, the idea we need later. And second, what is phosphorylation? It's nothing but the ATP synthesis. We are attaching a phosphate to ADP, adenosine diphosphate, to that adenosine diphosphate. When you attach a phosphate, you get ATP, adenosine triphosphate. Okay. So that's called as addition of phosphate. We call it as phosphorylation. So now to both words. Oxidation is removal of electrons, removal of hydrogen, addition of oxygen is called as oxidation. And addition of phosphate to ADP or anything else, we call it as phosphorylation. And basically, generally, when we use phosphorylation, it's nothing but ATP synthesis. Okay, understood that. Now, there are two types of phosphorylation. One we call it as substrate level phosphorylation. Other one is oxidative phosphorylation. We will go through both. First of all, what is substrate level phosphorylation? Here what happens is when a high energy compound like phosphoryl pyruvate, you know it glycolysis. In a high energy compound like phosphoryl pyruvate, when it undergoes the reaction, it will release some energy and converts to pyruvate. Glycolysis, the last step, phosphoryl pyruvate to pyruvate. At that time, what happens? That energy released is straight away used for ATP synthesis. So these type of reactions where the energy released from the high energy compound is straight away used for ATP synthesis. It's called as substrate level phosphorylation. And there we are not using we are not getting the help of electron transport chain. So energy released from a high energy compound is directly transferred to nucleoside diphosphate to form nucleoside triphosphate. Without the help of electron transport chain is called as substrate level phosphorylation. We should know some example. If it's a short answer, at least one or two examples you should know. You are familiar with the word. First one, glycolysis, we know. Phosphoryl pyruvate to pyruvate. Second one, the TCA cycle, succinyl-CoA to succinate. And another one, 1,3-bis-phosphoglyceride, 3-phosphoglyceride, again in glycolysis. So this is about substrate level phosphorylation. And these compounds, phosphoryl pyruvate, succinyl-CoA, 1,3-bis-phosphoglyceride, this can be called as high energy compound because it releases energy which is more than that is percent ATP. I'll come to that definition later when we say about high energy compounds. Okay. Now going to the other part. Oxidative phosphorylation to study, you should know a basic idea. In our body, when you have the food, the breakdown process takes place at 3 levels. Okay. One we call it as primary metabolism or digestion. You have the carbohydrate, you have starch, you have rice that contains starch. It digests, converts to glucose and getting absorbed. That is called as primary metabolism. And second one, secondary metabolism. After the absorption, it undergoes the catabolism process. That's called as secondary metabolism. Okay, so glucose converts to pyruvate, then to acetyl-CoA, acetyl-CoA undergoing TCA cycle, These all comes under secondary metabolism. Okay, in the secondary metabolism, one important point, what happens is that the electrons, high energy electrons and hydrogen ions from these compounds will be transferred to coenzymes. And when the coenzyme get the electrons and hydrogen, we call it as reduced coenzyme. Okay, what are coenzymes? You might have already undergone the revision. So what do you mean by coenzyme? Most of the students will say just the word non-protein part of an enzyme. But I'll tell you that's not the correct answer. The non-protein part of an enzyme is called as cofactor. When you say coenzyme, of course, that's not the same part, but exactly speaking, they are organic molecules, low molecular weight organic molecules, which are loosely bound to enzyme, what is essential for enzyme activity is called as coenzyme. I'll repeat once again because coenzyme is a short answer for you. Coenzymes are, if you are not able to catch up, you can share it in your live chat. Coenzymes are non-protein part. In fact, they are low molecular weight organic molecules. Coenzymes are low molecular weight organic molecules, which are loosely bound to enzyme that are important for enzyme activity. Okay. And then second point about coenzyme is what's the use of coenzyme? They are group transfer agents. Simple as that. They are group transfer agent. And mainly what are they? And the third point, most of the coenzymes are active form of vitamin B complex. Okay, active form of vitamin B complex. For example, our thymine pyrophosphate, NAD, NADP, FMN, FAD. These all are active form of vitamin B complex, and they are nothing but coenzymes. So I said 3 points about coenzyme definition and its main function, group transfer agents, and third, what are they? And there are 2 types of coenzyme, and one is there which help in the transfer of hydrogen. I said they are group transfer agent, so help in the transfer of hydrogen. And so that coenzyme that help in the transfer of hydrogen, they are NAD, NADP, FAD, and FMN. Okay, these 4. Now I'll coming back to my topic. During the catabolism process in the body of the biomolecules, the high energy electrons are transferred to that coenzymes. Which one? FAD or NAD and NAD plus, and convert to is reduced form FADH2 and NADH plus H plus. So that much happens in the secondary metabolism. Tertiary metabolism, or that's called as our oxidative phosphorylation, where what happens? We are going to say that. So now I'm coming to our important word today, oxidative phosphorylation. This is what happens here. I hope you can see these 2 lines here. It stands for the inner mitochondrial membrane. And we have the reduced coenzyme what are produced as the part of secondary metabolism. And that they are NADH and NADH plus H plus and FADH2. From there, these electrons will pass through these green colored things are some complexes, protein complexes. So when the electrons are passed through these protein complexes towards oxygen and convert to water, during this transfer of electrons, so electrons is transferred from one fellow to another. I said the removal of electron is called as oxidation. So the whole process, this which as I showed you is nothing but oxidation. At that point of time, during the oxidation, the energy release will be used for conversion of ATP to ADP plus PI to form to ATP, or for the synthesis of ATP. This coupled process of oxidation and this one, ADP plus PI, giving ATP is called as phosphorylation. So transfer of electrons or removal of electrons is oxidation. This is phosphorylation. So coupled process together, oxidative phosphorylation. So what is in basics? What is oxidative phosphorylation? Process of synthesis of ATP. Now we should say when it happens. during the transport of electrons. From whom? From the reduced coenzyme. To whom? To oxygen. Through whom? Through a set of protein complexes present where? The inner mitochondrial membrane. So those who have not written, we can write that. What is oxidative phosphorylation? No doubt, it's a process of ATP synthesis. No doubt. Now we say when? During the transport of electrons. When? During the transport of electrons. From whom? From reduced coenzyme. To whom? To oxygen. Through the protein complexes present in the inner mitochondrial membrane. This is called as oxidative phosphorylation. Hope you understood this definition clearly. It's a coupled process of oxidation and phosphorylation, no doubt. So you should say what is oxidation, what is phosphorylation. Now we'll study this process. So if the question is short answer question, short answer is about oxidative phosphorylation. You start with biological oxidation consists of three things: primary metabolism, secondary, and tertiary. Primary is digestion, secondary is catabolism, tertiary is oxidative phosphorylation. So say about each and say the definitions. And these, I said, the electrons are transported through some protein complexes. These protein complexes are called as electron transport chain. And the electron transport chain is a frequent question for the MBBS class examination. And for the BDS also 2 or 3 times it has been asked. So we'll have a look on that electron transport chain. So we said the definition about electron oxidative phosphorylation. Now we'll say about electron transport chain. Okay, we will go into that. What is electron transport chain? What's the definition for it? Just what we said of oxidative phosphorylation, we were concentrating on the process. Here, we are going to concentrate on the complexes. So we'll say about that. Electron transport chain are a set of protein complexes present in the inner mitochondrial membrane. The same thing, which are involved in oxidative phosphorylation. It's simple as that. Okay, so I repeat that. Those who are writing can add up that. We can add up that. They are the set of protein complexes present in the inner mitochondrial membrane. For what purpose? Which are involved in oxidative phosphorylation. It can be called as electron transport chain. Okay, it consists of, this electron transport chain consists of 4 fixed complexes and 2 mobile complexes. Mobile complexes, you just study the components, we just study the names, coenzyme Q or ubiquinone, and cytochrome C. We'll be telling about these fixed complexes. So those who are listening to this one, just have a pen with you. First of all, you draw the picture, schematic diagram on electron transport chain, then we will add up. I'll be going to the most important points about the electron transport chain, okay? So I'll be just showing you the pictures. So I hope you can draw this one. We'll add up in that. Take your paper in a horizontal manner. I can draw this one. The above picture, above 2 lines stands for outer mitochondrial membrane. And these 2 stands for inner mitochondrial membrane, where you can see 4 rectangular boxes. That stands for the complex 1, complex 2, complex 3, and complex 4. 2 circles, you can see, they are the mobile components. One mobile component is above the membrane, inner membrane. It is water soluble. Cytochrome C is water soluble. It cannot be inside. But the other mobile component, ubiquinone or coenzyme Q, what I've shown you in the pink color or purple color, that is inside. That's the lipid soluble part. So that is in the membrane only, in the inner membrane. And the last one is nothing but our complex 5 or ATP synthase complex. Okay. Now we'll draw something, we'll write. Okay. So complex 1, each complex says we'll study one by one. It's very easy. We are not supposed to then just say the word complex 1, 2, 3, 4. We are supposed to say the names. It's very easy to study. If you understand the concept, it will become very easy. Okay, first of all, about the complex 1. What the complex 1 do? Complex 1 is the protein complex that accept the electron from NADH plus H plus. Okay, this complex 1 contain mainly two components. One is FMN, another one is iron sulfur complex. So the electrons from NADH plus H plus first go and bind to the attached to FMN. It will become FMNH2. Then it will attach to iron sulfur complex, transferred to iron sulfur complex. It gets reduced. Then it will be given to our first mobile component, coenzyme Q. So now come to the point. So this is a complex which transfer electrons and hydrogen from NADH. Remove hydrogen from NADH. Removal of hydrogen is called as dehydrogenation. Enzyme name, dehydrogenase. So this complex 1 is removing electrons and hydrogen from the NADH. Hydrogen from the NADH. So the complex name is dehydrogenase. It's from NADH. So simple name, easy to study, NADH dehydrogenase complex. Why the word complex? Because it contains a number of components. So simple word, don't mug up. Instead you study in this way. This is the complex that transfer the hydrogen and electron from NADH. So the name transfer of hydrogen. So the word dehydrogenation, removal of hydrogen. So the word dehydrogenation. Enzyme name dehydrogenase. So NADH dehydrogenase complex. It contains mainly FMN, iron sulfur complex. And then what is function? Transfer the electron from NADH to the coenzyme Q. And second, one more function. During this process, this complex 1 acts as a proton pump. It pumps the electron from the matrix side towards the intermembrane space, the space between the outer membrane and the inner membrane. We call it as intermembrane space. It transfers 4 pumps 4 hydrogen ions. So we say 2 functions for complex 1. One, it transfers the electron from NADH and expressed to coenzyme Q. Second function, it transfers the hydrogen ions or pumps out 4 hydrogens from the matrix to the intermembrane space. Hope you understood this. And the enzyme, this complex 1 name, NADH dehydrogenase complex. Now we'll write that. You can make a tabular column in this way. All together, drawing on one paper and another page, you can do this one. Complex 1 is also called as NADH dehydrogenase complex. Major components are iron sulfur complex and FMN. There are 6 iron sulfur complex in that. It has 2 functions. One, transfer the electron from NADH to coenzyme Q. Another one, it acts as a proton pump. 4 protons are pumped out. I hope you know the word proton means H plus ions only. From matrix to intermembrane space. And who all are the inhibitors? That names you are supposed to by heart. Anyway, our anticonvulsant drug barbiturate is that. Some fish poison. Chlorpromazine is also another anticonvulsant. Pearcin is an antibiotic. These compounds act as an inhibitor. So our first complex of electron transport chain is over. Second step. So this is a complex through which the electrons from NADH enters. What about FADH2? Now come the other one. Mainly the complex 2. We'll draw this again. We'll go back to the other paper. From the succinate, I hope you remember this word succinate that is present in the one of the intermediate of TCA cycle. From the succinate, the electrons and hydrogen transferred to FAD to form FADH2. And from any FADH2 that is produced either through glycerol 3-phosphate or acyl-CoA, we will be studying beta oxidation, acyl-CoA dehydrogenase. The production of FADH2 is there. From that FADH2, this electron goes to some iron sulfur complex there. in the complex 2. And then the electrons are transferred to coenzyme Q. So that's about the complex 2. The complex 2 just function. The electrons from FADH2 enters the electron transport chain through complex 2. Similar one. Second point, contain iron sulfur complex. And how will you name it? From here itself. If complex 1 is called as NADH dehydrogenase complex, because it gets the electrons hydrogen from there, it helps in the removal of the hydrogen from NADH. So the name NADH dehydrogenase complex. So complex 2 is the one which helps in the removal of hydrogen from succinate. So the name can easily guess now. Succinate dehydrogenase complex. So it transfers the succinate. So complex 2 transfer or help in the removal of hydrogen from succinate or help the transfer of electrons from that. And then goes to FADH2, then goes to the coenzyme Q. So it's called as complex 2 is called as succinate dehydrogenase complex. One point is that it cannot pump any protons because during this process, the energy production is less so that it cannot act as a proton pump. So FADH2 will be producing, will not be able to release out. So when the electron from FADH2 enters the electron transport chain, at this point, at the complex 2 point, there won't be any proton pump. Understand one point there? When 4 protons are pumped, then 1 ATP can be produced. I'll be coming to that word later. Okay, now we'll write about complex 2 in our tabular column. Complex 2 is called as, you can say by yourself now, it's called as succinate dehydrogenase complex. Then, what are the components mainly? It contains iron sulfur complex. And then what it do? It transfer the electrons from succinate to coenzyme Q. Or you can write transfer electrons from NADH2 also, because, FADH2 also. FADH2 to coenzyme Q. Because FADH2 produced by the action of acyl-CoA dehydrogenase or glycerol 3-phosphate dehydrogenase, that also get transferred. That electron from that also get transferred to coenzyme Q through the complex 2. And mainly malonate, carboxyl, malonate, example for inhibition, carbon inhibition. You have studied already that. Yeah, that inhibit the complex 2. Okay. Now the electrons have gone to the coenzyme Q. Coenzyme Q you have written at the beginning itself. It's a lipid soluble compound. It's a mobile component. And we'll add up a word about coenzyme Q in the beginning itself. Up to this part, 2 electrons were given to him. But hereafter, electrons will be transferred as a single electrons, not as a pair. So we'll say, what's the function of coenzyme Q? Coenzyme Q will be acting as a bridge, as an intermediate between a 2 electron donor and a 1 electron acceptor. That's the thing you're supposed to know about coenzyme Q. It undergo a cycle inside itself, coenzyme Q cycle. Thereby, it acts as a bridge. You can, those who have not written, you can write that. Coenzyme Q acts as a bridge between the 2 electron donor and 1 electron acceptor. It's a mobile component. It's lipid soluble. So it moves. It comes over here to complex 1. Get that. Goes toward the complex 3. Now we'll study about the complex 3. What did the complex 3 do? Complex 3, first, it contains some complexes called as cytochrome B. You just draw this one. This picture itself will give you marks. You'll be able to effect the marks with the picture itself. It contains 2 things, cytochrome B, then iron sulfur complex and cytochrome C1. Then the electrons are given to cytochrome C. That's a water-soluble component that is present on the outer surface of inner mitochondrial membrane. Understand, one important point is that whenever there's a leak of cytochrome C, that leads to apoptosis, cell death. That's the importance of cytochrome C. So it's very much important. Fine. So what did the complex 3 do? Complex 3 gets the electron from coenzyme Q and give it to cytochrome C. Cytochrome C. And how will you name it? This is cytochrome C. How will you name it? So this complex 3 is the one which gives electron to cytochrome C, which gives electron to cytochrome C. So when you get electrons, it's getting reduced. So complex 3 is helping in the reduction of cytochrome C. So now it's easy for you to say the name. You can guess the name, which help in the reduction of cytochrome C. Reductase, cytochrome reductase. So complex 3 is called as cytochrome reductase, or common total word, cytochrome C reductase. Simple word, cytochrome reductase. Now we'll write about that. It contains, and one more function, during the electron transport, this also will act as a proton pump. 4 protons are pumped from the matrix to the intermembrane space. Okay, now we'll write this in tabular column. We'll fill up the tabular column there. Third complex is called as, it helps in the reduction of cytochrome C. So the word cytochrome reductase. And it contains 3 components. We have a cytochrome B, we have cytochrome C1, and we have iron sulfur complex. What did it do? Just now we said, it gets the electron from the mobile component, point sign Q, and give to cytochrome C, next component. Electron from point sign Q and transferred to cytochrome C. There it acts as a proton pump. When 4 protons are pumped out, that comes in back later. At that time, 1 ATP will be produced. Understand that. That basic concept you should know. And then, who all are the inhibitors of this one? Anti-mycin A, British anti-leucine. These are the 2 major inhibitors of complex 3. Now we'll go for the last one, complex 4. Complex 4. We'll go back from the cytochrome C. We go to complex 4. Complex 4 contains 2 copper atoms. We will not be showing the picture, but we should write that. 2 copper atoms and 2 heme atoms attached to cytochromes. Cytochrome is a heme containing protein. Please understand that. Only difference between hemoglobin and cytochrome is that, I'll repeat, only difference between cytochromes and hemoglobin. Heme. Cytochrome and heme is that. Both contain cytochromes and hemoglobin contain heme. In hemoglobin, iron will not be, normally iron will always be in Fe2 plus state. But in cytochrome, it will be reversibly converted from Fe2 plus to Fe3 plus. That's the difference. Okay, so this contains 2 heme atoms attached to cytochromes. We call it as cytochrome A and A3, or heme and heme A and heme A3. You can say it both ways. Okay, so we will write about heme alone or cytochrome alone, cytochrome A1 and cytochrome A3. But understand that before that there's a copper atom. 2 copper atoms are there. So electron from cytochrome C goes to cytochrome A1, cytochrome A3, then copper B, and then last, it goes back to the oxygen atom with the hydrogen and form water. So totally the electrons are transported now to the oxygen atom. I showed the first picture. The electrons move from the reduced point sign towards oxygen and forming water. At this time, this complex acts as a proton pump, but it pumps only 2 protons. 2 protons. So it will be equal to 0.5 ATP. Understand that way. So complex 4. Now we will repeat. What does the complex 4 do? It takes the electron from cytochrome C and gives to the oxygen. See, complex 3 was the one who gave electron to cytochrome C. So we called him cytochrome reductase. Hope you remember. Complex 3 was the one who provided the electrons, who gave electrons to cytochrome C. So it provided electrons to cytochrome C, it goes a reduction. So we use the word cytochrome C reductase or cytochrome reductase. Now complex 4 is the one who takes electron from cytochrome C. So naturally it is oxidation. So its name should be, come on. Hope you will be remembering now that term. Cytochrome oxidase. Complex 4 is called as cytochrome oxidase. Will contain these components. I hope you understood now. We will write about that. We'll complete our tabular column now. We complete our tabular column now. Fourth column, complex 4. Since it is removed electron from cytochrome C, it's called the cytochrome oxidase. It contains 2 copper atoms, copper A, copper B, and 2 cytochrome A1 and cytochrome A3, or that contain heme. So we use the word heme and heme A3. Don't get confused. You can write heme or cytochromes. I just wrote both because in some books will be given as cytochrome C, cytochrome A1 and A3. Some will be given as heme A and heme A3. So I don't want to make you confused. I just try to say both can be used. Okay. And then what is function? It also has 2 functions: transfer electron from cytochrome C to oxygen. And also one more function, it acts as a proton pump. And here comes our most poisonous compound. That's the inhibitor for it, which is the one: cyanide, carbon monoxide, hydrogen sulfide. These all inhibit the complex 4. Now, hope you understood. All our electron transport chain, the content, the main content is there in the one tabular column. If you have written all the things together, that's it. There's no, and this diagram will do everything. And then I didn't finish, but we have a little more. So we were here. I hope you understood these parts. Just trying to recollect the things. And then what happens? After this process, now listen to this one. Our electrons are transported, but the ATP synthesized not so far is not synthesized. Now what happened? When we compare to, think about our inner mitochondrial membrane. See this inner mitochondrial membrane. The inner membrane space and the matrix side. When compared to matrix side, the inner membrane space contains more hydrogen ion. So it's more acidic. Chemically, acidic is chemical gradient. Okay, there's a potential difference between these two. Not only that, then comes H plus ion, charges. Electrical charges will be more, plus ions are more on the inner membrane space. So because of this hydrogen pumping, when the protons or hydrogens are pumped out, because of which there's an electrochemical gradient is formed. And this is again a pressure, again a force. So far the electrons were pushing, so we call it as electron motive force. Now electron motive force, because of that the protons are pumped out. Now there comes a generated a proton motive force. With that force, these all protons are pumped back through complex 5 to the matrix. Complex 5 is also called as ATP synthase complex. ATP complex 5 is called as ATP synthase complex. Not only these 2H hydrogen ions, all the 10 are pumped back. When 4 protons are pumped back, 1 ATP will be produced. The next 4 protons, another ATP. When 2 protons are pumped, 0.5. So totally from the NADH, when the electrons are transported, please check it out. You get 2.5 ATPs. But from FADH2 transports, see, when the FADH electron from FADH is transported, only 6 hydrogen ions are pumped out. So they will be coming back and you get 1.5 ATPs. So that's why from NADH plus H plus you get 2.5 ATP, and from FADH2 you get 1.5 ATP. Okay, and this is called as, and this process, this phenomenon, we use the word chemiosmotic theory. Chemiosmotic theory. So when the electron transport chain is asked, you are supposed to complete the question using the word chemiosmotic theory. What it says, this word: transport of electron through the protein complexes. When the electrons are transported, as we said so far, that leads to the protons are pumped from the matrix to the intermembrane space. 4 protons from the complex 1. Complex 1 is also called as NADH dehydrogenase complex. Again, 4 protons by complex 3. What is its name? Cytochrome reductase. And 2 protons by complex 4. What is its name? cytochrome oxidation. These all, so because of that pumping, it generates an electrochemical gradient across the membrane, across the inner membrane, because of which a proton motive force is forms that pumps back the proton through the ATP synthase complex, the complex Y, that leads to ATP synthesis. This called as chemiosmotic theory. This theory that explains ATP synthesis during the transport of electrons through the complexes. Transport of electron through complexes is called as oxidation. At that time, ATP is synthesized. Because of the transport of electrons, protons are pumped out. That leads to, again, the proton motive force, or another word, electrochemical gradient is formed. That pumps back the proton through complex 5 or ATP synthase complex. That's helping ATP synthesis. Hope you understood this part. So electron oxidative phosphorylation. Then we said the components of electron transport chain. Then we said about ATP synthase complex. And then we are telling about chemiosmotic theory. No need to say much about ATP synthase complex here for you. We're not going so deeper. Just say a complex 5 is there. It's called as ATP synthase complex, through which the protons are pumped back. That help in the ATP synthesis. So that's all about the process of oxidative phosphorylation. Hope it's clear. With the tabular columns, everything is clear, I believe. Now we'll study about a word. Last exam, this was a short question. High energy compounds. What are these high energy compounds or energy rich compound? What is the definition? Any compound which has energy more than the ATP, or which can, on its breakdown, produce ATP on reaction straight away. So this word, having more energy than ATP, we call it as high energy compound. Compounds, the biological system, which on hydrolysis yield free energy equal to or greater than, or is missing there, that of ATP, is called as high energy compound. In the ATP, we have around minus 7.3 kilocalories per mole. For 1 mole of ATP, minus 7.3 kilocalories. So any compound which has either minus 7.3 kilocalories or more than that can be called as high energy compounds. That's the definition for it. Okay. And we'll find some examples. No doubt our ATP, GTP is a high energy compound. Cyclic GMP, cyclic AMP is a compound. And as I told you earlier, any compound on its reaction undergoes substitution phosphorylation. That can be called as high energy compound, or phosphoenol pyruvate, 1,3-bisphosphoglycerate, creatine phosphate, acetyl-CoA, acyl phosphate. These all are high energy compounds. And what's the use of it? What's the use of this high energy compound? Since it came as the last one question, let me complete that answer with some its function. What are the uses or functions? No doubt. One, for our movement, mechanical movements, for muscle contraction. Then active transport mechanism across the membrane, secretion of hormones, secretion of enzymes, mainly metabolism, anabolic processes, conjugation processes. Anabolism, we understand this way. For the glycogen synthesis, you need UDP glucose. That's the active form. So attachment of UDP only, with the help of UDP glucose is formed. That's a high energy intermediate compound. Okay. In that way, when we synthesize triglyceride in our body, CAMP acyl group will be attached. So, and then for conjugation, you know that bilirubin is conjugated with glucuronic acid. UDP glucuronic acid is attached for conjugation. So these are for this conjugation process, for the anabolism processes, we need it. So not only for muscle contraction, we need energy. we need for the active transport, we need for secretion, we need for metabolic processes, mainly anabolism, we need for conjugation. So this much about the high energy compound. Next is one more question from this electron transport chain topic. Another question that has been asked about uncoupler. What are uncouplers? Yes, we will recollect it. When the electrons were transported in oxidative phosphorylation, there are two processes. One is oxidation, other one is phosphorylation. Okay. So about oxidation, what is this transport of electrons from one complex to another? That is oxidation. You know it. And some chemicals, some endogenous compounds also, what happens is the transport of electrons will take place, but it will not allow to form a proton gradient. Why? Because that will increase the permeability for H plus ions. Instead of going through the complex Y, it goes to somebody else, somewhere else, so that the proton will come back. Because of which what happens? The ATP will not synthesize. So uncouplers are inhibitors of oxidative phosphorylation. It will not mind the oxidation, but it will prevent the phosphorylation process. So that's it. Agents that inhibit ATP synthesis, oxidative phosphorylation, by preventing the generation of proton gradient. When I taught you about the chemiosmotic theory, I use the word proton gradient. So usually the protons are pumped out, all the protons will be there, then the proton motive force forms. But here what happened? This fellow will increase, uncouplers will increase the permeability of inner mitochondrial membrane to protons, so that through some other way it goes inside, not through ATP synthesis complex. So ATP will not produce. So this is how it functions. This is the mechanism of action of uncoupler. It increases the permeability of inner mitochondrial membrane to protons. So in simpler words, it's an inhibitor of oxidative phosphorylation by preventing the proton gradient formation. How it increases the permeability of inner mitochondrial membrane to the protons. We have some physiological uncoupler. You know that some people, whenever they go to the chill places of Ooty or Kodaikanal, they won't feel much chill. Most of the people will shiver again. They won't be able to feel this one. Why? Because in them, they contain more brown adipose tissue, which contain a compound called as thermogenin. So one physiological uncoupler. Thermogenin inhibits what happened? It acts as inhibitor of oxidative phosphorylation. So what happened? Instead of ATP production, straight away heat will be released out. So thermogenin. Another one, thyroxine. In hyperthyroidism, please, you might have studied this thing already. Physiology. Hyperthyroidism. The patient will be having a high body temperature. Why? Because thyroxine is an uncoupler. So heat will be dissipated as, so energy will be dissipated as heat. Bilirubin. So please check the jaundice patients. In hyperbilirubinemia, they will be having a high temperature because it's an uncoupler. Some long chain free fatty acids is an uncoupler. We have some exogenous, that means some chemicals. 2,4-dinitrophenol, pentachlorophenol. And what's the effective uncoupler? As I said now, they are thermogenic effect. Heat is dissipated, energy is dissipated as heat. Thermogenic effect. Hope it's clear for you. Any doubts so far? In the live chat sections you can do. You can ask some questions. If you have any doubts, it can be answered. So we studied about, in general, not in general, in detail about the process, oxidative phosphorylation, substrate level phosphorylation, chemiosmotic theory, uncoupler, high energy compounds. Is it okay? You're able to understand now? Is it fine? Shall I go for the next topic? So this is one major topic that most of the students asked about oxidative phosphorylation and electron transport chain. Let me go for the next topic now. So same thing only, don't worry. The same thing I'm putting in this, we have already shown you. Leave it. Now about you want to study about beta oxidation. So I'll just start with oxidation of fatty acid. And the main repeated essay question. A repeated essay question. Beta oxidation. So for that, first of all, what is oxidation of fatty acid? Oxidation of fatty acid means breakdown, catabolism of fatty acids. As I told you earlier, any catabolic process is oxidation. So whenever you say oxidation, it's nothing but catabolism. Glycolysis we defined as oxidation of glucose, where glucose is broken down into pyruvate. So any breakdown, any catabolism, any breakdown process in the body is oxidation, simple as that. So now we'll go to, in that, the fatty acid, different types of fatty acid oxidations are there. The major one is beta oxidation. We have alpha, omega, peroxisomal, or chain fatty acid oxidation. Anyway, we'll be studying about beta oxidation and 1 or 2 sentences about or chain fatty acid oxidation. Because that also comes under and is beta oxidation only. Okay, for an essay on beta oxidation. On essay on beta oxidation, these are the things you are supposed to say. Suppose, right. For your examination, we should have a definition, we should have a site, and about the steps, the main 3 steps I have said: activation of fatty acid, transport, breakdown, energetics, how much energy is produced, regulation, disorders. Okay, we'll go one by one. Site alone, beta oxidation takes place in every cell except brain, because the fatty acid bound with albumin cannot cross the blood-brain barrier. So fatty acid will not be there, free fatty will not be there. So it cannot undergo beta oxidation. RBC don't have a mitochondria. Adrenaline blood also somewhat around that only. So in these 3 places, beta oxidation will not take place. Now we'll study about definition first. Okay, before that, the basic concept, this word. Think that you have some fat in your body. Whenever insulin is less, I want this thing later, but you say about the regulation part. Whenever you are in need of energy, when you don't have the glucose, enough amount of glucose, at that time, in the adipose tissue, the fat is nothing but triglyceride. Please not. Number of students make a mistake. They will always write the word fat for cholesterol. It's not. Fat means triglyceride, triacylglycerol. That means to a glycerol molecule, 3 fats are attached. That's called as fat. Cholesterol is totally different thing. So adipose tissue contain the fat. So whenever the glucagon level is more than insulin, that means whenever you are on starvation, whenever glucose is less, insulin will be less. At that time, glucagon will be more. That will activate an enzyme called as hormone sensitive lipase. HS stands for hormone sensitive. And an enzyme that breaks lipids, lipase or lipase. That is sensitive to hormones, so the word hormone sensitive lipase. That will be active in presence of our glucagon. If the glucagon is small, this will get activated and converts to glycerol and fatty acid. 3 fatty acid plus glycerol, we call as triacylglycerol. You understood that. Then this fatty acid is not soluble. So with the help of albumin-bound fatty acid, it goes to our tissues, mainly I'm just showing about the liver, comes to liver. Either it gets stored there or converts by the beta oxidation, converts to acetyl-CoA. Then this acetyl-CoA mainly, normally, it enters into TCA cycle. Mainly into TCA cycle. Or otherwise, if it's very high, if it's more, then it will convert to cholesterol also. And some amount, and in under severe starvations or uncontrolled diabetes mellitus, more amount of acetyl-CoA is there, then ketone bodies will be produced. This diagram will be helpful for you to say when ketonemia occurs, ketonuria, ketosis occurs, whenever glucagon is more fat breakdown, more fatty acid comes to liver, more beta oxidation, more ketone bodies. You can say that. Anyway, now I'm trying to say how the fat is produced. So this idea should be there in your basic concept, should be there in your mind. This happens whenever in our body, whenever you didn't have the food in time, whenever you are on starvation, whenever you are on fasting, this happens. So intermittent fasting will slowly reduce your adipose, the fat in your adipose tissues. Understand that. Okay. Now about beta oxidation. What is about the beta oxidation? Here, a long chain, around 18 carbon atoms. See this one, 18 carbon atoms are there. These all points stand for carbon atoms, okay? And last one, COOH. That's what CO2H here written. Now, what is beta oxidation? For the examination, I have seen a number of students just writing beta oxidation means oxidation at the beta position. The name came like that, but definition is not that. What is the definition for beta oxidation? Here comes the answer. See here what happens during the process. First, it cut here. In first cycle, it cut here. Remove the 2 carbon atoms as acetyl-CoA. Next, it cut here. It breaks. It breaks here. Again, 2 carbon atoms removed as acetyl-CoA. 2 carbon containing compound is acetyl. Then remaining, first 2 is removed, remaining 16. Then 2 is removed, remaining 14. Then 2 is removed as acetyl-CoA, then remaining 12. Then 2 is removed as acetyl-CoA, then 10. It goes like that, one by one. Sequentially, one by one, another word, sequentially. Removal of acetyl-CoA from which end? Carboxyl end. So, beta oxidation is a process of complete breakdown of fatty acid. That's our point. Where there's a sequential removal of 2 carbon units as acetyl-CoA. From which end? You have 2 ends. One carboxyl end, one methyl end. Carboxyl end, methyl end. From the carboxyl end, one by one, sequence it will remove the 2 carbon units as acetyl-CoA. So that's the definition for it. Beta oxidation. It's a process of breakdown of fatty acid. The first word, first part of the definition, you are familiar with. I told you, whenever you use the word oxidation, it's nothing but breakdown. Breakdown of fatty acid. Now comes the main point where there's a sequential, one by one, removal of 2 carbon units as acetyl-CoA from the carboxyl end. This is the definition for beta oxidation. Hope it's clear. Now, one more sentence you can write, as everybody writes. Why we use the word beta oxidation? Because during this process, this carbon atom, listen, this is the functional group, COH is functional group. The carbon atom next to it will be alpha. This is beta. To it, an oxygen is attached or get oxidized, simple word. So, because during this process, beta carbon atom get oxidized, it's known as, the process called as beta, or it's named as beta oxidation. So, definition is breakdown of fatty acid, whether there's a sequence of removal of 2 carbon units as acetyl-CoA. No doubt. Then why this name? Because oxidation takes place at the beta carbon atom. So, these two together give you a complete definition for your essay question. Have it in your mind, breakdown of 2 carbon units, oxidation of the beta carbon atom. I need it when I teach you about the oxidation process. Shall I go for the next slide? It's, as I told you earlier, mitochondria of almost all tissues except, I have already said, brain, RBC, and adrenal medulla, except in all other tissues, it takes place. Okay, if it is 18 carbon atom, then 9 acetyl-CoA. If it is 16, there will be 8 acetyl-CoA. Simple as that. You know it. Now, we'll come to the, so definition is over, site is over, steps. Steps mainly, as I told you, there are 3 major steps. First is activation of fatty acid. The simple word, fatty acid activation. What do? We attach a coenzyme A. Can you say what is coenzyme A? Coenzyme A is nothing but active form of pantothenic acid. OASH. SH group is active group in that. SH stands for thiol, okay? Thiol group. I need that word later. So first of all, just write this term alone. So we have second is transport of fatty acid, third is breakdown process. We'll stay one by one. First one, activation of fatty acid. Here what happens? Similar words, fatty acid plus coenzyme A give fatty acyl CoA. Here, what happened? During this process, one ATP is there, adenosine triphosphate. From that, two phosphates are removed. So two high energy bonds are removed and produce AMP plus PPI, inorganic pyrophosphate. PPI stands for I for inorganic, PP stands for pyrophosphate, means two phosphate. This is equivalent to utilization of two high energy bonds, so two ATPs, equivalent to conversion of 2 ATP into 2 ADP. Okay, so remember that we need later when we study the energetics. So that's the first step in the beta oxidation step. And this alone takes place in the cytoplasm of the cells. From this, if a short chain or medium chain, that itself easily is permeable to the mitochondrial membrane, easily go inside. Okay, so this takes place in the cytoplasm, two energy bonds are utilized. Enzyme name, simpler word, you can say it this way. Fatty acyl CoA is synthesized. So the word synthesis, and ATP is utilized. So we use the word synthetase. If any compound is produced without the utilization of ATP, then we use the enzyme name synthase. Whenever ATP is utilized for a synthesis, we use a general word synthetase. Okay, acyl CoA synthetase, or common word fatty acyl CoA synthetase. Some book use the word thiokinase also, but this is more easy for you to remember. Acyl CoA or fatty acyl CoA synthetase is a better word. Okay. Second, transport to mitochondria. As I told you, the short chain fatty acid and medium chain fatty acid can easily are permeable to the inner mitochondrial membrane. But the long chain fatty acids are not permeable. So it needs a taxi to go inside. It needs a car, and we name it as carnitine. Simple. Carnitine is a compound. There is a translocase, a transporter for the transport of carnitine or its substituents. Substituted carnitines. We use the word carnitine translocase, common word. So the carnitine cannot go inside, can go inside, but acyl CoA cannot. Okay, what is carnitine? Name is beta hydroxy. If beta position, we have a hydrogen group, and the gamma position 3 methyl amino group, methyl group attached to an amino group. So ammonium, gamma trimethyl ammonium butyrate. Butyric acid is the one. Carnitine is butyric acid, where at the beta position there's a hydrogen group, at the gamma position 3 methyl group is attached to the amino group. Okay, it carries the long chain activated fatty acid. We'll show how. We'll make it more clear. Instead of this picture, we'll go make it clear more this one. Let this be our, just for understanding sake, our inner mitochondrial membrane. We have a translocase enzyme there, translocase protein. And at the outer side of inner mitochondrial membrane, please note, we have an enzyme called as CAT1, carnitine acyl T for transferase. Carnitine acyl transferase 1. Understand that. What it do? It will transfer the acyl group to the carnitine and form acyl carnitine. Acyl CoA plus carnitine will combine together in presence of carnitine 1, forms acyl carnitine. This acyl carnitine can go inside. That car can go inside, can go. Translocase is there. Already go, now went inside. So this dotted line stands for it transported to the matrix side. This is the matrix side, this is the cytoplasm side. There, as soon as it comes into the mitochondria, in the inside part of the inner mitochondrial membrane, we have CAT2, carnitine acyl transferase 2. What do you do? Still convert back this acyl carnitine to carnitine and acyl CoA. So acyl CoA was not able to go inside. So it is converted to acyl carnitine. Then acyl carnitine goes inside. After that, it goes back to acyl CoA and carbon. Then When one carnitine goes out, one acyl carnitine comes in. This is what it meant by. And this shuttle, we call it as carnitine shuttle. The same thing, another picture in a common textbook. Our transporter, on the outer side of the membrane, you have the carnitine, acyl carnitine, carnitine acyl transferase 1. Your acyl-CoA, fatty acid plus carnitine give acyl carnitine that goes inside acyl carnitine, then with the help of another CAD2, carnitine acyl transferase 2, convert back to carnitine and acyl-CoA. And this translocase deficiency or carnitine deficiency, what happened? The fatty acid will not go inside, so you won't get energy. Muscle get totally exhausted. So in fasting conditions, so muscle cramps occurs suddenly in fasting conditions. Whenever we do exercise, high fat diet. So whenever you have, instead of having glucose, if you have more amount of fatty acid or fat, then the muscles will not get energy if there's a deficiency of translocase. That's one clinical importance. Okay. Now comes to the next part, the main steps of beta oxidations. We'll study about that. First one. See, I have drawn a picture here. Don't worry about this picture. I have written the names also there. Fatty acyl-CoA. Think about palmitic acid, 16 carbon containing compound. And alpha position, beta position, just for your sake of understanding. And we call it as acyl-CoA, fatty acyl-CoA. First step, our name word was, what is the beta oxidation? Oxidation of at the beta position. So at the beta position, you want to have a oxygen should be attached. Think it does, okay? But what happened? Carbon atom has all the 4 valencies. All the valencies are saturated now. So if you want to attach oxygen, somebody should be checked down. So it has 4 rooms. Carbon atom contains 4 rooms. So all the 4 carbon atoms. So 4 rooms of carbon atoms are full now. So we are asking them to check out. So we are removing 2 hydrogens from there. The first step, removal of hydrogen from that. And what happened? A double bond formation. And hydrogen one to this side and one to this side. So the word trans-enoyl-CoA. Trans-enoyl-CoA. And there's a removal of hydrogen atom from the acyl-CoA. So acyl-CoA dehydrogenase. Similar that the first step, acyl-CoA dehydrogenase. And that can draw here from that acyl, from the fatty acid, hydrogen atom is removed. So the enzyme name dehydrogenase, FAD accepted and converts to FADH2. We have already studied from FADH2 1.5 can be produced. Please note that. Okay. The first piece over fatty acyl-CoA to trans-enoyl-CoA or enoyl-CoA, simple word. Remember this word F, then E. Next. Now I want to attach oxygen. Now the room is vacant. At the beta position carbon atom, room is vacant. I want to attach oxygen, but I'm sorry, I can't attach oxygen as such. So we'll add water, OH and H. So the beta position, OH will be attached. Alpha position, H will be attached. So what happened? Now hydrogen will be, oxygen will be attached. So at the beta position, oxygen is attached by addition of water. Addition of water is hydration. So the enzyme name, hydratase. Enzyme name, hydratase. Can you name this one? To the acyl-CoA at the beta position, or it is attached. So the word beta hydroxy acyl-CoA. Very good. Beta hydroxy acyl-CoA. The word beta hydroxy acyl-CoA is produced. Now, I said I want an oxygen atom at the beta position, not OH. So what I do, again, I'll ask him to check out. One H from O and one H, this one. These 2 H will be removed. Again, removal of hydrogen. So the enzyme name, dehydrogenase. So this time, it will be accepted by NAD+, converts to NADH. And then only oxygen will be there. double bond O. So the word that C double bond O is called as keto. At which position? Beta position. So the name beta keto. Beta keto acyl CoA. And the enzyme name, of course, from the beta hydroxy acyl CoA removing hydrogen. So again, dehydrogenase. Beta hydroxy acyl CoA dehydrogenase to form beta keto acyl CoA. I'll just show once again. Beta hydroxy acyl CoA, removal of hydrogen, and form beta keto acyl CoA. So our definition part, the second sentence is fine now. Now the first sentence, removal of 2 carbon units. You are going to cut here. 2 carbons are removed. Using one again a coenzyme A. As I said, coenzyme A contain SH group. SH is thiol. Breaking using thiol. When you break a compound using water, we use the word hydrolysis and the enzyme name hydrolase. You're going to cut here with the thiol, SH group. So the enzyme name thiolase. Using thiolase, we cut that. You get again a fatty acyl CoA plus acetyl CoA. That acyl CoA will be having 2 carbon atom less. That's it. Plus acetyl CoA is 2 carbon atom. So 16 become 14 and 2 is removed as acetyl CoA. In this way it happens. So during this process, during this one step itself, 1 NADH plus H plus and 1 FADH is produced. So 1.5 plus 2.5. 4 ATPs are produced in one cycle itself. Remember that. And as you know, acetyl CoA can enter into TCS cycle. From acetyl CoA, how many ATPs? From one TCS cycle? If you don't remember, please study it this way. TCA, T, and again, our number is also started with T, 10. TCS cycle produced 10 ATPs. One acetyl CoA, when it undergo TCS cycle, Sadiga cycle, enters TCS cycle and catabolism occurs, it produces 10 ATPs. Have in your mind. So this is about beta oxidation steps. And then again, it undergo these steps are repeated until it's completely degraded. That's about beta oxidation steps. Okay, so here comes some students can be able to understand the chemistry and be able to say that. Some students will struggle in studying the chemistry part. So we'll try to study something else. We start with the fatty acid. We started with the fatty acid here, right? So in one Western country, there is a dance called as fungi elephant dance. And the fungi elephant have kingi, K for kingi. So study in this way. First compound is fatty acyl CoA. Second is enoyl CoA. So F fatty acid fungi elephant E enoyl CoA. Then have kingi, K for keto. Kingi lex always, A for acyl, again acyl CoA. Fungi elephant have kingi lex always. Using that mnemonics, you'll be able to, if you don't want to study the chemistry part, study it that way. Fungi elephant have kingi lex always. F stands for fatty acyl CoA. Then E for enoyl CoA. Have H hydroxy acyl CoA. Then kingi K keto acyl CoA. Then lex, there's nothing like that. Then again always A acyl CoA, again fatty acyl CoA and acetyl CoA. Okay, so study in that way also. You can study the chemistry ways. I try to teach you the concept behind it, the word from the beta oxidation, how to have the oxidation at the beta position. I just try to teach you in that way. or otherwise using the mnemonics. Okay, now we come to the part energetics. Energetics. This is a carb. See, always we use example as palmitic acid, 16 carbon atoms. So as I told you earlier, in every cycle, one FADH and one NADH is produced. FADH2 and one NADH plus H plus are produced. Okay, that means from one cycle, 4 ATPs. Okay, so totally how many cycles? For complete breakdown, check it out now. 16 carbon atoms, each breakdown produce 2. So we think 16 by 2, 8. It's not like that. See here, we removed 1, 14 remaining. Then 2, again remove 2, 12 remaining. Then remove 2, 10 remaining. Then remove 2, 8 remaining. Then remove 2, 6. Then 4, then 4. After that, 1. So totally how many cycles? So we always say n by 2 minus 1 cycle. Other words, n by 2 minus 1 cycle. For a complete breakdown, you need n by 2 minus 1 cycle. Each cycle produces 4 ATPs. And the end product is acetyl-CoA. How many acetyl-CoA? N by 2 acetyl-CoA. If we have these two basic concepts, and from one acetyl-CoA, when it enters the TCA cycle, how many ATP? Just now I said TCAT 10. 10 ATPs from one acetyl-CoA. If we have this thing, energetics is over. Energetics. This is what all about. Number of cycles required 7. Each cycle produces 4. So 74, 28. Then number of acetyl-CoA produce 16 by 2, 8. Number of ATPs from one acetyl-CoA is 10. Number of acetyl-CoA, so number of ATPs from 8 acetyl-CoA, 80. Totally we get 108. It's not over yet. During, I asked for the net gain, net yield. During activation, we utilize 2 ATP. Number of ATPs utilized, 2 for activation. So net gain, 106. Hope you understood this part. So 2 basic concepts, n by 2 minus 1 cycle and n by 2 acetyl-CoA. If you understand these two, then it becomes an easy thing. So this much amount of energy from one palmitic acid. From glucose, you get 32 ATPs. See, 3 times more than 3 times, right? 106 ATPs are produced from palmitic acid on the fatty acid oxidation. And about, next comes, I'm sorry, we'll just leave that. Regulation. How the process is regulated. Just now I said, see, in the beginning I said of a concept. Whenever the glucagon level is more fat breaks, more fatty acid comes to the liver or tissues and more breakdown. The first thing that happens about regulation of beta oxidation is that first regulation is by availability of free fatty acid. Whenever the glucagon-insulin ratio, we always take that. Glucagon, when you say glucagon is more, insulin is less. When the glucagon-insulin ratio is more, that means under starvation conditions or whenever diabetes mellitus condition, at that time what happened? Glucagon is more. Whenever this ratio is more hormone sensitive lipase will be activated, more free fatty acid level, more beta oxidation. This is the first idea. That's why I showed the concept first, the breakdown of fatty acid there. Second is about the rate-limiting enzyme, CAT1. Mitochondrial uptake of fatty acid using the CAT1. CAT1 is an allosteric enzyme. It is activated. Malonyl-CoA is the inhibitor of CAT1. So these are the 2 major ways by which the process is regulated. When the malonyl-CoA is more chance of fatty acid synthesis. That's what. The starting compound for fatty acid synthesis. So these two are about the regulation. So an essay question, we said about the definition, side we said, steps we said, energetics, regulation part, how the process is controlled. That means fatty acid oxidation will not take place always. Whenever it's favorable condition, whenever the glucose ratio is high, then only the hormones and the lipase breaks and adipose tissue from the triglycerides and the adipose tissue breaks. Then only you get the fatty acid, then only the beta oxidation. Understand that. Okay, leave that. Some, at least one or two disorders. If the fatty acyl-CoA dehydrogenase enzyme is deficient in the newborn baby, what happened? For them, breast milk is the most important diet. It contains more amount of medium chain fatty acid. So if the medium chain fatty acyl-CoA dehydrogenase, if it is deficient, what will happen? It cannot utilize the fatty acids in the breast milk. All the glucose will be over, hypoglycemia occurs, and it may lead to sudden death, sudden infant death syndrome. That's one disorder related to beta oxidation. Defect in medium chain acyl-CoA dehydrogenase occurs in infants. Suddenly the glucose level decreases, goes hypoglycemia. And leave over that. Whenever the beta oxidation will not occur, the omega oxidation occurs. That's why the word dicarboxylic aciduria occurs in this case. So one thing, just name it. You are supposed to know the word SIDS, sudden infant death syndrome, deficiency of medium chain acyl-CoA dehydrogenase. One more, this is what I will say. It's called as Jamaican vomiting sickness or Jamaican vomiting disease. Here what happens? A fruit, a K fruit, if it is untried, what happens? It contains a compound called hypoglycine that will inhibit the acyl-CoA dehydrogenase. So that will lead to, again, hypoglycemia. Whenever the hypoglycemia occurs, there will be vomiting. So this is what about Jamaican vomiting disease. Okay, so two disorders related to our beta oxidation. Now, one thing, let me say, so much we have studied, we'll write one extra point. Okay, what is it all about? It's all about beta oxidation of our odd chain fatty acid. You have studied in different places there. As a part of B12 function, as a part of our steps of gluconeogenesis, as a source of gluconeogenesis. And as a question there, so it comes in different parts. It will be a revision for you. Since you have already studied that, we'll just add up here. Oxidation of odd chain fatty acid. Please write this much alone. Odd chain fatty acid oxidation, that's the difference. The last product will be propionyl-CoA. Beta oxidation of our even chain fatty acids produce acetyl-CoA fully, but here this difference is production of propionyl-CoA. Okay, so your beta oxidation essay question as a last part, as separate one, you add this point. See, after all, you are trying to impress your examiners through the examination to get the marks. So write some extra points. And with this one, you are studying there. Beta oxidation of odd chain fatty acids produce propionyl-CoA. This step you have already studied, or you are supposed to study that. Propionyl-CoA, with the help of propionyl-CoA carboxylase convert to methyl malonyl-CoA. This is an example for our ligase enzyme and function of biotic. Ligase enzyme classification, the sixth class, ligase enzyme. Propionyl-CoA carboxylase or acetyl-CoA carboxylase. You are adding carbon dioxide group, ligase. And next step, you also are studying this one, L-methyl malonyl-CoA to succinyl-CoA. This is only two functions for B12. One is this function. Function of B12 is an important question. There, see, this is one function. L-methyl malonyl-CoA to succinyl-CoA using B12. And this succinyl-CoA and then the gluconeogenesis. See, and why I start this thing? Because we have mainly four sources for gluconeogenesis. One source is our lactate. Second source is glucogenic amino acid. Third source is odd chain fatty acid. And fourth source is glycerol. You are studying this one. In gluconeogenesis, in essay question, you are supposed to write this part. If you study there, you can add up here, no, easily. And you get extra mark also. The examiner will get impressed about you when you write about beta oxidation part. This is an extra point. Anyway, you are studying for the gluconiensis. Now we can add up here too. So that's all about our beta oxidation. And today's last question is about HDL metabolism. A lot of students ask me for this one, a revision on HDL metabolism. Revision on HDL metabolism. Please go through this one, HDL metabolism. Here, as you know, the word lipoprotein. Lipids cannot be transported as such because they are hydrophobic in the blood. In that blood is aqueous medium. It cannot be transported. So for a transport, we need somebody else's help. It's nothing but the protein. So a combination of non-covalent aggregation of lipids and proteins called as lipoprotein. One of the major lipoprotein is HDL. You know about all the lipoproteins. I'm just showing you a one tabular column on the chylomicron, VLDL, LDL, HDL. I'm just going to concentrate on HDL alone. HDL helps in the reverse transport of cholesterol. That means it transports cholesterol from tissue to liver. Okay. So to study about HDL metabolism, first we will study, listen very carefully. First, we will study the proteins or enzymes involved in HDL metabolism. First of all, we will say the function. As I told you, reverse transport of cholesterol. Why the word reverse transport? See, in all other things, just our chylomicron, VLDL, LDL, all were transporting towards tissue. This is the only one from tissues to liver. So the reverse transport of cholesterol. It transports cholesterol. Only that. Not only that, it provides C2 and E for other lipoproteins. So it's a reservoir for it. It's one more function for HDL. Act as a reservoir, serve as a reservoir of C2 and E. That's one thing. Next one is about ApoA1. Major protein HDL metabolism is ApoA1. It's an integrated protein. It cannot be transferred to any other one. ApoA1. ApoA1 is a major apolipoprotein. What it do? It activates LCAT. We'll study about LCAT now. Third one, I'll teach you that in another slide. Another one, ABCA1, ATP binding cassette transporter A1. What is function? From the tissue, cholesterol should be transferred out, pushed out. That is done by ABCA1, helping the efflux of cholesterol from the peripheral tissues. Next, hepatic lipase. Some amount of triglyceride is present in the HDL that is digested, hydrolyzed using hepatic lipase. Okay. Now comes LCAT. Lecithin is a phospholipid, okay, which contain PUFA at the second position and cholesterol. Now, what LCAT do? What is LCAT? Lecithin cholesterol acyl transferase. From the name itself, it's a transferase. What it transfer? Acyl means whom? The PUFA. From whom to whom? From lecithin to cholesterol. This word, LCAT or lecithin cholesterol acyl transferase, will transfer. See now, this PUFA, unsaturated PUFA, from lecithin to cholesterol to form cholesterol ester and lysolecithin. So this action of LCAT. This LCAT is activated by ApoA1. This LCAT is present in the blood, the serum. When the HDL comes, it binds it to it. Okay, we'll come to that word. So we said about LCAT now. Function of LCAT helps in the esterification of cholesterol with PUFA. So now understand, PUFA is important for HDL metabolism. PUFA or essential fatty acid. This is one of the functions about essential fatty acid. And next protein, CETP, cholesterol ester transfer protein. What do you do? In the HDL, if there is a lot of cholesterol ester, then some amount of cholesterol will be transferred, will be shifted to either VLD or LDL by CETP, cholesterol ester transfer protein. In exchange of triglycerides, he won't give by himself simply. When HDL gives that, it gets back to triglyceride. This triglyceride is digested using hepatic lipase. And then last, the uptake of cholesterol and our HDL by liver using a SRB1, scavenger receptor B1. Okay, so these are the proteins involved in HDL metabolism. Now, with a very simpler picture, I'll try to say the functions. First, when you study HDL metabolism, understand this way. See, in town area and all, the kitchen waste, for taking the kitchen waste, there the people comes from the municipal town area. Municipal workers are there. They comes with a 2 cans, the trolley or a small vehicle. Maybe 2 or 3 cans will be there. Now, as soon as they comes, they will sound horn. At that time, what happened? From the kitchen waste will be somebody else will be taking that to that fellow. It will be sorted out and they keep the can in the cans. Once the can is full, it will be given to some other bigger cans, bigger vehicle. Again, goes back to the other houses collected. The same way HDL do. Now think, in the liver or in the intestine, the HDL is nascent, HDL is produced. The picture, see that. And of course, ApoA2. So when it's produced, it will be having ApoA1. That we call as nascent HDL. It is disc shaped. And it gets ApoE and ApoC in circulations. And then what happened? It has ApoA1. ApoA1 will activate the LCAT, LCAT, and LCAT will get attached to the nascent HDL. Now the nascent HDL goes to tissues. It is like our municipal workers, it makes on. Now what happens from the peripheral cells? You have the cholesterol inside. So somebody else is taking the cholesterol. Now ABCA1 push the cholesterol out. So he's giving the cholesterol to the HDL, nascent HDL by ABCA1, ATP binding cassette A1, protein A1. Shifted the cholesterol. Okay. And then that cholesterol as such will not be taken up by the HDL. It will be sorted out. When you give some kitchen waste, it will be sorted out. Same way, what happens? This cholesterol will be converted to cholesterol ester using the LCAT, lecithin cholesterol cell transferase. Lecithin is present on the outer surface of our HDL. It's a phospholipid. It is there. From there, the PUFA will be attached to cholesterol. Then the cholesterol ester enter into the HDL. Now it becomes circular. The disc-shaped nascent HDL is discoid shape. It becomes circular. Cholesterol ester rich. Now this HDL is called as HDL3, cholesterol ester rich. Either it can be taken up by the liver, or what happens when it is become rich in cholesterol ester, it cannot get other cholesterol from others. So what happened? This cholesterol ester will be given to the VLDL. Okay, you take it. It's given to VLDL with the help of cholesterol ester transfer protein. In return, it gets triglycerides. Now, triglyceride-rich HDL is called as HDL2. Now, this HDL2 is taken up by liver. First, the triglycerides are digested with the help of hepatic lipase. The remaining cholesterol in HDL is taken up with the help of SRB1. This is very simplest picture available now regarding the HDL metabolism. I'll repeat once again. New HDL is produced from liver or intestine. It is nascent. It is disc-shaped, discoid shape. It contains ApoA1. Once it comes in circulation, the ApoA1 activates LCAT, and LCAT from the blood, it will attach to it. As soon as it reaches the tissue capillaries from the peripheral cells, the cholesterol efflux occurs with the help of ABCA1, ATP binding cassette transporter A1. Efflux happens. Cholesterol as such will not be taken up. Cholesterol will combine with the PUFA from the lecithin itself, which is present on the outer membrane of HDL, outer surface of HDL. And then what happened? It becomes cholesterol ester. That cholesterol ester goes inside. It is hydrophobic, goes inside. Now it becomes spherical shape. This cholesterol ester rich HDL is called as HDL3. Simple as that. Then, when cholesterol ester is is high, is more. It cannot get other cholesterol from other peripheral tissues, but it want to collect it. So what do you do? This cholesterol ester some amount will be given to the VLDL using CETP, cholesterol ester transfer protein. In return, it gets triacylglycerol. This triacylglycerol rich HDL is called as HDL2. Then as soon as it goes towards liver, HDL2, the triglycerides will be digested, hydrolyzed using hepatic lipase, and the remaining is taken up with the help of SRB1. This is all about HDL metabolism. So if an HDL metabolism is asked, we can say about lipoprotein in general. They say the function, two major functions of HDL, reverse transport of cholesterol and reservoir of ApoC2 and D, because it provides for the VLDL and chylomicrons. Can say that. Then say about the metabolism. Synthesis from the liver or intestines, digestion is HDL is discoid shape, gets LCAT, activated by ApoA1, takes up the cholesterol from peripheral tissue with the help of ABCA1, converts to cholesterol ester, HDL3 is formed. From there, it's transferred towards VLDL, transferred to VLDL with the help of CETP. In return, it gets triglycerides, then converts to HDL2, then taken up by the liver. That's all about HDL metabolism. The same thing are in other words now. I'm just same thing only. This is what the intestine and liver produce. HDL is this shape. Cholesterol is there in the tissues. Afflex using ABCA1. LCAT bind to it. The cholesterol is taken up, converts to cholesterol ester, and taken up by the this one HDL3 cholesterol ester. Then from the HDL3 either can be taken up by the liver, or HDL3 converts to HDL2. What happened to the VLDL? Give us with the help of CETP. Cholesterol ester given. In return, it gets triglycerides. Convert back to HDL. Converts to HDL2. Taken up by the liver with the help of SRB1 and hepatic lipase break it. Okay, same thing in words only. And it will be there in that. Later on, if you want to write these points, you can write down. Cholesterol from the cell is transferred by HDL with the ABCA1. ApoA1 of HDL activate LCAT track acidification cholesterol with PUFA. Now comes the function of PUFA. PUFA helps in HDL metabolism and thereby reduces the level of cholesterol. So it's called as good cholesterol. HDL is called as good cholesterol because as soon as the cholesterol is brought to the liver, it can be excreted out through the bile. So the level of cholesterol decreases with the help of HDL metabolism. The first HDL is called as HDL3, then mature HDL, either taken up by the ApoA1 with the help of SRB1 mediated process, or some amount of cholesterol is transferred to VLDL, LDL using CETP in exchange for triglyceride. This triglyceride particular HDL is called as HDL2. They are taken up by the liver. Triglycerides are digested with the help of hepatic triglyceride lipase and taken up with the help of SRB1. And I put here, LDL is called as bad cholesterol, HDL is called as good cholesterol. Why? Because HDL will reduce the cholesterol level. And thereby, it is anti-atherogenic because it transports the cholesterol towards liver, thereby helping the excretion. So it's having a hypocholesterolemic effect, anti-atherogenic. Instead, LDL, what it do? It takes the cholesterol towards tissue, causes accumulation of cholesterol. So it's called as. So it's atherogenic. LDL is atherogenic. So it's called as bad cholesterol. So these words also, this also can be added up. Thank you for your patient listening. Maybe in the next class, as you have already said some questions from amino acid metabolism, we will cover that topics in the next class. At that time, I'll give you the other important questions too. Thank you for your patient listening. Hope it's clear. Any doubts can be asked in our WhatsApp group too. I'll always there to help you out. Any doubts you can ask at that time. Thank you.